Hello, everyone. I'm glad to be part of this year's conference. My name is Henry Matov. I'm a PhD candidate at Curtin University. In this presentation, I'm going to share what we have found so far in terms of how university chemistry students explored an enzyme reaction in immersive virtual reality. This study is part of a research project led by Associate Professor Mihai Wang and is funded by the Australian Research Council. Chemistry educators are aware of students' difficulties in visualizing 3D molecular structures. To support students' visualization, educators use physical models such as the one shown in the figure, or molecular models on 2D computer screens. However, the challenge of visualization persists when dealing with complex molecules such as proteins, since we cannot easily adopt these visualization tools. Therefore, it is worthwhile to investigate the possibilities of alternative visualization platforms such as immersive virtual reality. This is what our research team at Curtin University is doing. Immersive virtual reality is an advanced 3D visualization tool. Once learners put on headsets, their view of the physical world disappears. The learners feel they are teleported to a virtual yet realistic looking molecular world. In immersive virtual reality, what I will be calling IVR, learners can explore complex molecular structures in 3 dimensions as well as manipulate them freely, as shown in the figure. This could help them visualize complex molecular structures better to improve their conceptual understanding. Learners can also share the VR space and interact with one another, allowing them to learn collaboratively. In this study, we investigate how learners explore the IVR environment from multiple angles. One is their spatial exploration, that is how learners explore the virtual space. 2 is their social exploration, how they interact with one another to explore the virtual space. And 3 is their conceptual exploration, how learners are developing their scientific understanding in IVR. Target concepts in this study were molecular polarity and shape in the context of an enzyme reaction. The concepts were chosen because they are fundamental to understanding physical and chemical properties of molecules. For example, these concepts are key in understanding enzyme reactions. As a case study, our team designed an IVR application to allow learners explore how the enzyme acetylcholinesterase breaks down the neurotransmitter acetylcholine. We chose this context because enzymes are complex molecules that students find difficult to visualize. The research question was, how do students interact spatially, socially, and conceptually within IVR to improve their understanding of an enzyme substrate reaction? Participants were 66 first and second year university chemistry students. All the students were chemistry majors and had gone through 2 other IVR activities prior to the current activity. To provide an opportunity for collaborative learning, the students were paired to go through the activity. Our data collection protocol involved the pre and post interviews as well as actual IVR sessions. In the pre-interview, we asked learners about their understanding of enzyme reactions, and we asked them to draw diagrams to illustrate their ideas. In the post-interview, we asked learners to challenge their initial ideas about the reaction and to comment about their learning experience. Each pair of students spent about 1.5 hours to go through the whole activity. We collected data in form of videos, audios, student diagrams, and screen captures of what the students were seeing in IVR. The learning activity involved 6 tasks. I will refer to these later. 1 was constructing the substrate molecule and its electron density map. 2 was exploring different models of representation of the enzyme structure. 3 was zooming into the active site of the protein. 4 was to decide on the best orientation of the substrate at the entrance of the enzyme. 5 was to explore the catalytic chamber and its electron density. And 6 was to drag and position the substrate in the catalytic chamber in a way that would allow the catalytic reaction to take place. As noted earlier, we investigated how students explored in the 3D environment in 3 ways. One was the spatial aspect, which parts of the protein the learners explored and for how long. The social aspect, that is how they decided on who manipulates the molecules and how the learners talk to one another to build consensus. Third was the conceptual aspect, what kind of target concepts the learners discovered and the level at which they discussed them. The schematic diagram on the right highlights the relevant features of the protein environment they explored, that is the regions, the shapes, and the electron densities. In this diagram, red represents the electron-rich areas, while blue represents electron-poor areas. The target chemistry concepts were the shape of the entrance, the electron density of the entrance, the shape of the passageway, shape of the catalytic chamber, electron density of the catalytic chamber, and reactivity of acetylcholine. In terms of the students' spatial explorations, we observed that while in the IVR environment, students perceive the environment as real. For example, when moving in and out of the protein, they duck their heads down to avoid hitting the protein cloud. The figure on the right is an example of students' behavior while walking in and out of the protein. Also, when a student wanted to walk into the protein, but realized that the substrate molecule was in the way, they always walked around it as if it were a solid obstacle. Students also differed in the way they explored the environment. Some students explored the environment extensively, while others did not. In the next slides, I'm going to share our preliminary findings from 2 pairs of students who showed distinctive exploration behavior. The focus of the analysis was on the last 4 tasks, which required students to walk into the protein. That is, tasks 3 to 6. Even though the students were required to complete the same tasks, these 2 pairs of students differed in the way they explored the 3D space. The graphs here show the amount of time the students spent exploring each region in the protein. In tasks 3 and 4, Andrea and John showed hesitation to enter the protein, and they spent a significant amount of time at the entrance. However, task 5 and task 6 specifically asked the students to go into the protein. In task 5, because Andrea was entering the chamber for the first time, the peers spent a long time within the chamber. In task 6, the students quickly walked into the protein, placed the substrate there, and walked out. On the other hand, Tammy and Alfred were more extensive in their exploration. The students walked into the catalytic chamber to explore the features there in all the tasks. Because it is a 3D environment, the way students explored the protein environment was tightly linked to their social and conceptual explorations. Socially, the dads differed in the way peers communicated with one another to build consensus. For Andrea and John, the peers maintained different perspectives for most of the time. Each peer stood on a different side of the IVR space. As you can see from the figure, Andrea is on the left, John is on the right. Andrea is focusing on the red regions in the entrance, while John is focusing on the shape of the entrance. So because of the difference in perspectives, consensus building was very superficial with statements like, okay, we can just go with that. On the other hand, Tammy and Alfred made more efforts to get an idea of what the peer was saying. They frequently swapped their positions in IVR or stood beside one another to discuss their ideas and reach a consensus. The figure shows 2 students standing next to one another and having similar views. As shown in the diagram, the number of times peers made efforts to assume similar perspectives was significantly higher for Tamia and Alfred compared to Andrea and John. Also, because the students had one enzyme and one substrate molecule, we looked at how the students shared these resources to generate ideas during the tasks. As shown in the table, Andrea and John exchanged control of the molecules 8 times over the 4 learning activities. On the other hand, Tammy and Alfred exchanged control 14 times. For Andrea and John, the molecules were manipulated by John for most of the time. Therefore, he contributed more to the conceptual talk. On the other hand, Tammy and Alfred frequently took turns at moving or manipulating the molecules. The peers had almost equal contributions to the talk. In terms of the nature of their conceptual discussion, the target chemistry concepts were discussed at different levels in both dyads. In some cases, the concepts were simply mentioned without further elaboration. For example, so this area is blue. The student is referring to the area being electron poor, but does not say that this area is blue. In other cases, the elaboration was superficial and entirely based on the features of the environment. For example, so the blue would probably go to the red area, right? In this case, the student is talking about the electron poor region going with the electron rich region. However, they don't say that. In this case, we coded the level of discussion as low-level elaboration. High-level elaboration was when students integrated relevant prior knowledge in the discussion with clear relevance to the task at hand. For example, so that's the electron rich. So that will go with here, the electron poor part of acetylcholine. When we compared the dads in terms of their conceptual exploration, we found that Tammy and Alfred, who explored the environment more extensively, also engaged in more productive conceptual discussion. The graphs here show the number of speaking turns the peers engaged in for each target concept and the level of conceptual discussion. Tammy and Alfred discussed more target chemistry concepts, and their discussion was more elaborate than that of Andrea and John. In most cases, Andrea and John focused on simple mention of the target concepts or low-level elaboration. On the other hand, Tammy and Alfred had more elaborate responses, more elaborate discussions. As chemistry educators, we stress the value of 3D visualization in learning, and we design tools to help students visualize molecular structures and properties. In this study, our team designed a realistic-looking 3D environment with the hope that students would readily explore it to build their understanding of 2 concepts, that was molecular polarity and structure. Learners interacted spatially, socially, and conceptually in the 3D environment in different ways to obtain the learning outcomes. Some students felt hesitant to explore this realistic-looking 3D environment, while others extensively explored the 3D environment and discovered chemistry concepts that they would otherwise not have discovered. Because of the differences in the students' interaction patterns in IVR, there is need for researchers to further investigate how students interact with IVR to improve their chemistry understanding. This is a work in progress. Hopefully, we shall come back to share with you our findings at a later conference. Here are the references used in this presentation. To find out more about our IVR applications, please type Immersive Chemistry VR in YouTube. We have several videos of all the applications. You can also just send us an email at this address. Thank you for your attention.